Okay, hello everyone, and welcome to the sixth webinar of BioMeca. Today we are going to speak about skin regeneration, and more precisely, we are going to talk about the identification of cell subpopulations of the basal layer of the epidermis and how it is possible to detect them by both protein and mechanical markers. So to begin, I would like to introduce myself. I am Jean-André LAPART, and I am lab manager at BioMeca. I am also in charge of some projects in research and development. BioMeca is specialized in biophysics and in life sciences, such as cosmetic and pharmaceutical field, and we do our best to make the link between biophysics and the significance in biology from biological samples. Our innovation team is composed of different parts. The first one is to develop the new innovative field in order to provide new solutions. The second one is the hardware part and 3D printing in order to develop new products and prototypes. And at last, we listen to you and design with you the appropriate protocol that suits your projects to provide customized services. So now let's go to the heart of the matter. Epidermis is composed of four cell layers, with at its base the basal layer that includes proliferative and undifferentiated keratinocytes. Then these cells will undergo a vertical differentiation to form suprabasal layers of terminally differentiated cells. These cells will be progressively enucleated to form squames that will be shed from the skin surface. The turnover of cells of the epidermis is performed in about 14 days, and it requires 14 further days to complete the process of desquamation. Skin homeostasis compensates the loss of cells that are lost during the process of desquamation by the generation of new cells, which is ensured by the basal layer. Skin is confronted to many situations that can alter or modify its morphology, its composition, or its biological functions. Among these situations, we can find skin aging that includes intrinsic and extrinsic aging, but also skin wound. So during skin aging, the capacity of regeneration of skin is decreased, and this results in a decrease in the renewal of the epidermis. This also results in the reduction of the thickness of the epidermis and also by a flattening of the dermal-epidermal junctions. Skin aging leads to a decrease in the number of basal cells in the epidermis, and this is due to a reduction in the proliferation rate of basal cells compared to young skin. So this is important because this means that the epidermis, and more precisely basal layers of cells, undergo important modifications during skin aging. In the case of a tissue damage, the skin has to be repaired as quick as possible to prevent excessive loss of blood and infection. Wound healing occurs through four distinct but overlapping phases, which are the hemostasis, the inflammation, the migration or proliferation, and the remodeling phase. The hemostasis phase begins immediately after skin injury and leads to a formation of a blood clot which stops the hemorrhage and triggers the recruitment of immune cells in order to prevent some infection and to trigger and to activate the inflammatory response. The migration and proliferation phase coordinates the epidermal re-epithelialization in order to fulfill the epidermis in new cells. At last, the phase of maturation or remodeling consists in the removal of cells that are no longer necessary at the site of the injury, and this phase is also involved in the remodeling of the extracellular matrix. This phase of migration and proliferation is important and involves a pool of specific and plastic cells that are required for cell proliferation and the re-epithelialization of the epidermis. Thus, it is important to maintain an adapted skin homeostasis throughout life to allow skin to answer as best as possible to the biological modification. However, what are the cells that are necessary for skin homeostasis and skin repair, and where are they located? Cell turnover and epidermal repair are in part ensured by epidermal stem cells. A lot of studies have been led in mouse and in human for several decades in order to identify potential stem cells in epidermis. Even if it is still in debate, many evidences have been found in favor of this population of cells in epidermis. These stem cells are able to self-renewal and to proliferate. While it has been well studied and characterized in mouse, functional studies lack in human skin. However, it has been shown that stem cells are located either in the intrafollicular epidermis or in the hair follicle. On this scheme are indicated some proteins that are considered as markers of stem cells. Most of the stem cells in mouse have been found to be located in the hair follicle in three distinct regions, which are the bulge, the isthmus, and the infunibular. In the different markers specific to skin stem cells in mouse are colored in red, while in black are indicated the markers of putative stem cells in human skin. Some studies in human skin identified two populations of cells, which are the intrafollicular stem cells and transient amplifying progenitor, also called committed progenitor. The hypothesis being that these intrafollicular stem cells, which are quiescent cells in vivo with low cycling, are able to asymmetrically divide to form transient amplifying progenitor or committed progenitor that will proliferate and then will divide and differentiate to form the suprabasal layer of the epidermis. A big difference between mouse and human skin is the density in hair follicles in the skin, which is much higher in the case of the mouse compared to human skin. Hence, while it is obvious that these stem cells could participate in the maintenance of homeostasis of the epidermis in mouse and in skin repair, it is less obvious in human and reinforces the interest for the research of intrafollicular stem cells in human. Some studies have revealed that in mouse, epidermis homeostasis is mainly performed by both intrafollicular stem cells and intrafollicular committed progenitors. And this was demonstrated by the use of mouse skin model where mouse are lacking from hair follicles and where epidermis, this mouse managed to reconstruct an intact epidermis, but with a little delay of time. So these results indicate that stem cells from hair follicle in mouse are not essential for the maintenance of the homeostasis in skin, but only participate. As regards the wound healing process, some functional analysis in mouse using lineage tracing have been performed, and from these studies, it has been demonstrated that all stem cells from populations from all origins contribute to the processes of wound healing in mouse skin. However, it has been shown that these different populations of stem cells participate from a different manner to the processes of wound healing, as it has been shown that intrafollicular stem cells and committed progenitor, intrafollicular stem cells in red and committed progenitor in blue only participate to the wound healing at short term after the time of injury, whereas stem cells from hair follicle participate in a longer term during wound healing process, as these populations of stem cells are identified and detected several weeks after the time of injury. As I previously said, experiments in mouse and the lower density in hair follicle in human skin reinforces the hypothesis that interfollicular stem cells in human are responsible for the epidermal homeostasis and repair in human skin. Hence, it has been proposed that in human skin, stem cells are located along the basal layer above the dermal epidermal junction, here stained by marked by a staining by collagen 7, and this stem cell could be located either at the zone at the dermal papilla or at the bottom of the ridge ridge. Indeed, some markers have been identified to be specifically expressed in cells located at the dermal papilla area, or to be expressed only in cells located in the ridge ridge. Some other markers have been found to be expressed widely along the basal layer, but present some enrichment in cells located in the dermal papilla of the interfollicular epidermis. These markers include beta-1 and alpha-6 integrin, which are described as two of the first stem cell markers in epidermis. And as you can see on these images, beta-1 and alpha-6 integrin are widely expressed by basal cells along the basal layer, but we can detect some accumulation, some enrichment of this protein in cells located at the dermal papilla area. At the opposite, some proteins, such as the MCSP, LRIG1, and delta-1 proteins, are only located in cells, only expressed in cells located at the level of the dermal papilla. So here in red for MCSP, in green that for LRIG1 that overlap with MCSP, and delta-1 here in white. At the opposite, some proteins, such as the keratin 15, are located and enriched in cells located in the ridge ridge area, and sometimes we can detect weak expression in cells located in dermal papilla, but the cells that more express these proteins are located in ridge ridge. So based on the expression of these proteins, this indicates that basal cells from the interfollicular epidermis possess heterogeneous cell population. In order to determine the potential of stemness of this cell population, several approaches have been led. Obviously, in human, we are technically limited in vivo by ethical reasons, and genetic studies such as lineage tracing are difficult to perform in human skin as it is made in mouse. So the main approaches to identify the potential of stemness of this cell population include the analysis of protein pattern after submission of the skin to a stress or during the comparison between a young and an older skin. Some approaches consist in the extraction of cells from the epidermis, or basal cells for the epidermis, for in vitro culture, which is widely used for this type of experiment. And a more trending approach is the analysis at the single cell level for the identification of specific gene expression or protein expression by specific cell subpopulation of basal layers. Technically, in vitro, a lot of approaches or experiments have consisted in testing the capacity of proliferation of these different cell subpopulations by a clonal growth assay, or to test the capacity of adhesion of a cell to a matrix, the capacity of migration of a cells, of course, the analysis of the expression of some proteins depending on the cell population, and of course, some genetic manipulations, such as knockdown or overexpression of a protein, to identify its function. From these experiments, some arguments have pleaded in favor of the stem cell potential of certain cell subpopulations, especially cells that express high level of beta-1 integrin and cells that are able to form clonal growth in culture for a long time. Another argument is that cells that are relatively quiescent in the epidermis, in the intrafollicular epidermis, because they does not or less express the Ki-67 proteins, which is a marker for cell proliferation. And then the last argument is that stem cells have the capacity to fastly adhere to a substrate than non-stem cells. As the opposite, cells with low expression of beta-1 integrin that form abortive clone of terminally differentiated cells in culture are described as non-stem cells. These non-stem cells possess a higher proportion of proliferate in the intrafollicular epidermis in basal cells, and these cells have a lower capacity to adhere to a substrate compared to what is described for stem cell population. Some functional analyses have been performed in order to identify the function of these proteins that could identify stem cell population. So it has been shown that in the interfollicular epidermis, cells that express the MCSP protein also express higher level of beta-1 integrin, as you can see here in the cluster in yellow, compared to cells that do not express MCSP protein, but only express the beta-1 integrin, as you can see in the cluster of cells only in red. In the same manner, cells that expressed in the intrafollicular epidermis MCSP does not express or less express the Ki-67 marker protein in red, and this indicates that these cells expressing MCSP proteins are quite quiescent cells. After cell extraction of basal cells culture and in vitro culture, it has been shown that cell clones that express both the MCSP protein and beta-1 integrin have a higher capacity of proliferation in time compared to cell clones that does not express MCSP protein, but only the beta-1 integrin that form abortive and differentiated cell clones. Concerning the LYG1 proteins, some functional analysis of knockdown and overexpression have been shown that the knockdown of the LYG1 protein increased cell proliferation compared to the control, and as the opposite, the overexpression here of the LYG1 protein lead to a reduction of cell proliferation compared to the control. So this indicates that the protein LYG1 is involved in the regulation of cell proliferation. By staining of epidermal stem cells tissue, it has been shown that cells that express the LYG1 protein here at the level of the dermal papillae are not positive for or less positive for the Ki-67 protein, which is labeled in red, meaning that LYG1 promotes stem cell quiescence, and this has also been demonstrated by the fact that LYG1 regulates the AGF-MYC signaling pathway, which is involved in cell proliferation. So by inhibiting this signaling pathway, this confirms that LYG1 is involved in the regulation of cell proliferation. In addition, it has been shown that both NCSP and LYG1 protein are lost or mislocated in skin psoriatic lesions, where I remind you that in the case of psoriasis, we have an hyperproliferation of keratinocytes, and that these two proteins are lost or mislocalized, mislocated in this kind of skin lesions. At last, the protein delta-1, which is also expressed at the level of dermal papillae, have been found to inhibit the notch signaling pathway, which is involved in cell differentiation, and so by negatively regulate this signaling, this prevents cell differentiation in cells located in the dermal papillae. So all these data support the idea that cells in dermal papillae that express these proteins possess a strong stem cell potential. The dermal-epidermal junction present undulations, and these undulations are progressively flattened during aging, as illustrated on these images and on this graph. during aging. The dermal epidermal junction is not the only one structure to undergo changes at the level of the basal layer. Indeed, some studies have demonstrated that the expression of both MCSP in red, integrin beta 1 in red, and keratin 15 proteins was reduced during aging, as you can see here on these images and on the graph on the side, where during aging we have a reduction of the expression of these markers at the level of the dermal papillae. In the case of MCSP and beta integrin is widely expressed in the intrafollicular epidermal. And in the case of keratin 15, we have a decrease in cell expression at the level of the root ridge. These modifications are not only detected in intrinsic aging, but also in the case of the extrinsic aging, such as the exposition of the skin to the UV irradiation. So in this study, they compared skin from different body area, the abdomen here and the face here, and the authors made the postulate that skin face is more exposed to skin to UV, than skin from abdomen. So from this imaging, it's interesting to note that sun-exposed skin possess less MCSP and keratin 15 expression than less exposed skin to UV, as you can see on these peaks, and this whatever the age. Indeed, if we compare both skin from 60 years old and 30 years old, we can see we can still detect a decrease of the MCSP proteins or keratin 15 proteins in face skin, corresponding that meaning that skin that are most exposed to UV lead to a reduction of these two proteins. So this indicates that during skin aging, and skin aging in general affects epidermis, notably by leading to a reduction in stem cell population or putative stem cell population. So this reduction in stem cell population, as you see here on these images, is concomitant with a flattening of the dermal epidermal junction, is concomitant with a reduction in the epidermal thickness, and in the decrease in the removal of the epidermal and wound healing process. And this is due to probably due to a decrease in epidermal putative pool of stem cells. It is known that stem cells are organized in niches that are favorable to their maintenance. In addition, it has been shown that stem cells could have a specific or different mechanical profile compared to other cells, and this could be due to the environment that surrounds the niche of stem cells, but can also be due to the cellular cohesion, which could be different, the topography of the substrate, here the dermal epidermal junction, or the state of differentiation of the different cell subpopulations that we analyze. Hence, we would like to determine if the different cell population of the basal layer of the epidermis possess specific mechanical properties or not, and if it yes, what is the impact of aging on these mechanical properties? So to answer to this question, we perform specific measurements by atomic force microscopy on cells positive for the MCSP expression or negative for the expression of MCSP, and these cells are either located, and these IFM measurements were only performed at the level of cells in dermal papillae or cells located in a rich range area. So briefly, the atomic force microscopy is a technique of visualization that is based on the interaction between a thin tip placed on at the end of the flexible cantilever and the sample. Different types of measurements can be performed by IFM. So for example here, by scanning the surface of a sample, we can, it's possible after reconstruction to make a topographic image from the surface of the sample. We can also determine mechanical properties such as the stiffness of a sample by locally indenting the sample at different area, and at the end we can extract the general stiffness of the sample, or we are able to generate some stiffness map. This is an animation about the principle of AFM, where we can see that the AFM tip enters in contact in a non-invasive manner with the sample, and this enables, through the generation of force indentation curve, to determine the stiffness of the sample. So by AFM, we analyze the stiffness of cells expressing or not the MCSP proteins, and we have identified that cells expressing the MCSP proteins here have a stiffness stiffness profile which is superior to the one of cells located in the wheat range that does not express the MCSP protein. We perform the same analysis in older skin and have identified the same type of pattern of stiffness between these two populations of cells. Thus, subpopulations of basal cells from the basal layer of the epidermis possess specific mechanical properties. In addition, the difference in cell stiffness between these two populations of cells tends to decrease during aging. In this, we can see that the ratio of stiffness between these two populations of cells decrease during aging. So this indicates that during aging, we have an alteration of mechanical properties of the cells of the basal layer, as illustrated by this decrease of the ratio of the stiffness. To conclude, we have seen that skin is morphologically altered during aging. This affects both the homeostasis and the wound healing process of the skin, as stem cells are involved in these two processes. We have identified that putative stem cells located on dermal papillae possess specific mechanical properties, and during aging, we have a decrease in the expression of putative stem cell marker and in their mechanical properties. This leads to the basal layer to tend to be homogeneous in terms of cells population as compared with a younger skin, where we can distinctly identify several cell subpopulation based on the expression of several markers, or based on the mechanical properties of the different cell subpopulation. This type of measurement by AFM microscopy, combined with the monitoring of expression of putative stem cell markers, is a powerful tool for characterizing the effect of active ingredients targeting at an anti-aging and regenerating functions. Don't hesitate to send some email if you would like some precision about some specific topics that I have told during this presentation, or about the mechanical properties of stem cells. I'd like to thank you for your attention, and see you next time for another webinar from BioMeca team. Thanks a lot. Bye.